Fantastic. Thank you very much indeed. Thank you for inviting me and asking me to do this. I always enjoy these sort of things, and it's quite a nice time in the evening. Um, After a beautiful day, I have to say. My name's Peter Reynolds. My primary job is I'm a paediatrician, and I've always really focused on babies. I also work at the Royal Holloway University of London, and I'm clinical lead for the neonatal network for Kent, Surrey and Sussex. And tonight I'm going to talk about probiotics and gut health, and what I think parents need to know. The talk will be slightly technical in places, but I'm hoping you'll bear with me, and I'll walk you through it. And obviously, this is being done for Down Syndrome UK, which is a fantastic organisation, as you all know. I'm therefore acutely aware, of course, that many of you will have children or family with Down syndrome, but also that you will have children or family who don't have Down syndrome. And I think there are parts of this talk that I hope will be relevant for you as well. Okay, so what I want to be able to do is, okay, so statement of interests. Down Syndrome UK have organised this webinar, and I've recently received remuneration for various activities from the companies listed below. Okay, so what am I going to do this evening? Well, I'm going to give an overview of probiotics. I'm going to discuss the importance of having a healthy gut microbiome and what a microbiome is in the newborn and infant period in particular. I'm going to go through some of the evidence for the use of probiotics in children. The evidence is exploded over the last 2 to 3 years in particular. And then I'll finish off with describing what we know at the moment about probiotics and what some of the evidence is for probiotics in the gut in Down syndrome. In terms of terminologies, I want to give you a couple of examples of terminologies of things that are important. So SCFAs are the first one that you may hear about. These are short chain fatty acids, and these are the compounds that are released from indigestible foods by probiotics when they ferment them, and they release things like acetic acid, propionic acid, and butyric acid, which are really important chemicals that are widely involved in the body in making vitamins, providing energy, making good fats, but also interact with the immune system and the nervous system as well. So short chain fatty acids, SCFAs for short, are important. And in the early stages of life, when babies are receiving breast milk, if they're receiving breast milk, HMOs are human milk oligosaccharides that you may have come across. And these are sugars, but they're not designed to provide calories. They are purely designed to feed probiotic bacteria, which is a remarkable bit of evolution. Breast milk has about 4,000 different compounds in it, and HMOs are just some of them. But yeah, they're non-nutritional sugars. Okay, so what are probiotics? Well, we hit confusion with the very first slide because the WHO define probiotics are live microorganisms which, when ingested in adequate amounts, confer a health benefit to the host. Unfortunately, the European Union and the UK and the FDA in the States and others don't agree because they don't believe that a food can confer a health benefit. And therefore, over the years, you will have seen adverts by Activia and others being regularly pulled by the Advertising Standards Authority because they're not allowed to use the word probiotic in case the public get confused. I don't know if I'm breaking the law by giving this webinar, but let's hope not. But the term probiotics is technically not allowed because the public may get confused about it. So there is already confusion right from the get-go. What is a microbiome? So we often refer to the gut microbiome, but as humans, we have bacteria inside us living very happily and actually giving us a benefit all over the place. So a microbiome simply refers to a community of bacteria or microbes which live in a particular environment, and they are dynamic communities, so they're constantly interacting with each other. It's not as if they're ignoring each other, and some of the compounds they release when they're metabolizing will help or hinder other bacteria as well. And we have microbiomes in our gut, so that's probably the biggest one, but in our skin, in our lungs, and other places as well. And microbiomes are all around us in animals and soils and plants and seas and everywhere. So we have evolved side by side with bacteria, and their existence are very much crucial to our systems and our normal development and survival. And dysbiosis is the term that we use when there is a disruption to the normal balance in the microbiome. Because happens in a dysbiosis is the normal healthy composition and balance of microbes and the metabolic activity that undertakes in those microbiomes become abnormal, and species that may be considered to be helpful and dominant become underrepresented, and species that are normally suppressed or even absent can become established or increase. And I'll give you an example. Yeah, you may have come across the phenomenon that when you have antibiotics, some people are prone to getting thrush. And this is a great example of where the antibiotics kill off the healthy microbiome, and the Candida fungus, which is normally suppressed, becomes dominant and results in thrush after the use of antibiotics. So that's a good example of a dysbiosis. But as I say, what we do need and what we know is that a healthy gut with a diverse microbiome, a really good range of microbes in the gut, which consist of so-called healthy bacteria, the term we're not allowed to say, is essential for good long-term human health. And this is a diagram of all the various things that that probiotics can give, and a nice healthy gut can provide for us. Oh, sorry, I've gone through that a bit quickly. And you can see there are the SCFAs promoting fat storage, angiogenesis at the bottom there. This is normal blood vessel development. And we've got vitamins, we've got immune storage, and we've got central nervous system involvement. So this very large community of microbes, which reaches into the trillions in the adult gut, has profound effects on many of the normal functions in humans, and important functions as well. So probiotics, as we refer to them, are microbial preparations that we can take or give to people, or we can give them to animals as well, which are designed to colonize or recolonize the gut with healthy organisms and go back to suppressing those harmful organisms or potentially harmful organisms, and improve the dysbiosis of the gut. So that's really why we use probiotics and what probiotics are there to do. And probiotics help the gut to mature and stay healthy in babies and younger children. It can improve gut maturity and barrier and the function. But they also have these complex interactions with fundamental systems in our bodies, the nervous system and the brain, the endocrine and metabolics and systems as well. And they modulate the immune system as well. So there are a number of probiotics that are considered to be anti-inflammatory, for example. And they appear to be related to the emergence of allergic responses as well. And there are considerable concerns that over the last 20, 30 years, we've seen remarkable increases in in allergies in children, which seems to have been mirrored by increasing use of antibiotics and increasing delivery by caesarean section, for example. And again, these probiotics release these all-important short-chain fatty acids, amongst other things as well. And we know quite a lot about this because of the explosion in research that's happened. And I've provided some references which you can stick into Google if you want to read a little bit more. But there are now established links in adults where gut dysbiosis is linked with obesity and type 2 diabetes. And these were observations that were made many, many years ago and have stood the test of time. There are links with inflammatory bowel disease. There are links with allergies, autoimmune conditions, with colorectal cancers, and other neurodevelopmental problems or neurological problems such as Parkinson's and Alzheimer's. So these are some of the key morbidities, sicknesses, diseases that affect adults, all of which have been linked to having an abnormal gut. Now, of course, sometimes there's a little bit of chicken and egg, which came first, but it's an extraordinary range of conditions which have been tested scientifically and have found to be associated with a dysbiosis. And in children, as I mentioned, things like asthma and eczema have been associated strongly with the dysbiosis, again, inflammatory bowel disease, irritable bowel syndrome, but also the emergence of neurodevelopmental problems such as autistic spectrum disorder and ADHD. And again, we've seen a rise in these conditions, some of which has been attributed to better diagnosis and better reporting, but may also be linked to wider use of antibiotics and changes in modes of delivery. And again, in children and teenagers, the onset of obesity and metabolic problems may be linked back to the health of their gut right from birth. And from birth, dysbiosis has some profound effects in premature babies who suffer dysbiosis from an immature gut and exposure to a lot of antibiotics. It causes severe problems. Necrotizing enterocolitis is a rare but sometimes fatal condition. Babies are more vulnerable to getting infections. They're more vulnerable to having gastroesophageal reflux and feed intolerance. And they may start to carry antibiotic-resistant gut bacteria, which is considered to be a huge health problem globally. And by 2030, it's estimated that antibiotic resistance is going to be the biggest killer of human beings on the planet. So these are globally important issues. Okay, so that's really the context of probiotics and the gut and some of the conditions that they're strongly associated with. So let's delve into a little bit more about what probiotics we're talking about. So the most important thing is if we're going to take or give probiotics to another human is that they have to be safe. We definitely want them to be effective. We want them to do something, but they definitely have to be safe because these are live bacteria, and if it's the wrong sort of bacteria, they can get into the bloodstream, and bacterial sepsis can make people extremely unwell. So for human use, probiotics need to be quite carefully controlled. They should always be genetically characterized. It's no good just people saying, well, this is on the label, therefore that's what's in it. Until you actually genetically prove what's in it, you don't actually know. And there are a number of studies where people have gone to health food shops and analyzed what's in probiotic preparations, and perhaps unsurprisingly, they're not exactly what you would want. They need to be devoid of antibiotic resistance genes. What we don't want to do is give microbes to somebody and find that they carry antibiotic resistance as a result. We want them to be metabolically active, of course, because that's how they might provide a benefit. Ideally, they should be designed for use in the target population Remarkably, at the moment, probiotics seem to be quite a generally beneficial population, but there is plenty of interest in trying to design target bacteria for target conditions. And whilst that's not in general use at the moment, it's very much an aspiration. Commercial preparations need to be manufactured to good standards, to current GMP, and they need to be batch tested, and they need to make sure they haven't got contamination in them. They need to be stable. A lot of probiotics are labeled with the counts at manufacturer, but very few are labeled with a guarantee that the counts will still be there at the end of the shelf life. And it's no good manufacturing a probiotic if 10 minutes later all the bacteria are dead. So they need to be stable, they need to be easy to store, and they need to be easy to give. And of course, ideally, what you want to see is a background where they have been given widely, they are safe, and they are effective. And that takes time for that evidence to accumulate. So the most widely known, I think, probiotic class are probably bifidobacteria. I guess that's arguable, but bifidobacteria are hugely important because they colonize the human gut very, very early on. So especially in babies who are fed breast milk, the gut, the lower gut in particular, is full of bifidobacteria, and there are over 70 recognized species and 10 subspecies. And that's when I'm talking about the diversity that needs to be there. And that's what we want to see, lots and lots of species all interacting with each other. Only a few of them can use the HMOs, the human milk oligosaccharides, as their sole carbon source. And I've listed them there. And these are considered to be the champion colonizers of the human gut. Particular Bifidobacterium infantis is considered to be particularly important. Just waiting for my slide to move on. Let's just try that one there. Okay. So Bifidobacterium infantis is called the champion colonizer of the neonatal gut. It can use those HMOs. It produces lactic acid, which is quite good for fighting off infections. And it is able, therefore, to gain a competitive advantage against other bacteria. And it has anti-inflammatory effects. Bifidobacterium bifidin is another early colonizer. Lots of evidence that it helps to develop and mature the immune system and can directly influence the activity of immune cells. Again, it is anti-inflammatory and can reduce inflammation in the gut as well. So those are a bit about bifidobacteria. The other big group you may have come across are lactobacilli. So lactobacillus tends to occupy the small bowel and a bit higher up in the gut. So they're found in the mouth, digestive tracts, but also in the vagina as well. And that's one of the mechanisms where babies may become colonized with lactobacillus during vaginal birth. And again, they colonize the gut early, they metabolize lactose and HMOs, and they help to get rid of bilirubin. So when babies are jaundiced, a nice healthy development of lactobacilli in the first few days helps to get rid of that jaundice. Again, they have anti-inflammatory effects. Again, they're particularly good at helping to reduce the likelihood of infections. And lactobacillus seems to be also present in human milk. So there's a direct route where mothers who are breastfeeding are able to provide lactobacillus. And this seems to be something that, again, has evolved through many, many years of evolution. And there are other probiotics that are used. Streptococcus is a non-pathogenic bacteria which has anti-inflammatory effects. And in fact, Saccharomyces boulardii is a yeast. It's a fungus, which again has some probiotic effects as well. Those are the main groups of probiotics. Now, I'm going to talk about the newborn microbiome, the gut microbiome, because in the newborn infant, whilst it might not be completely sterile at birth, and there's some evidence that it might not be, it certainly is fragile. And I think we probably need to look after it a bit better than we currently do. As I mentioned earlier, the human gut, as it evolves, is a complex ecosystem. There's over 1,000 bacterial species in a normal human gut. And in adults, when it's mature, it contains between 30 and 400 trillion microorganisms. The gut contains more DNA than the rest of the cells in our body. We're actually outnumbered by our own bacteria, which is a slightly unpleasant thought, but we'll move on. But the neonatal gut does not have that diversity or those numbers. It has a limited amount and a limited diversity of bacteria. And previously, people have talked about the neonatal gut being sterile. Actually, as I said, it isn't, but that's because the numbers were quite small and on older scientific testing methods, quite difficult to detect. And then the gut becomes colonized in a vaginal birth. You might get some vertical transfer of the vaginal microbiome, and then you get skin-to-skin contact. Caesarean sections are sterile because they're operations, but there is skin-to-skin after the delivery. And then there is further colonization through breast milk and, of course, the environment that is around the baby as well. Now, I've mentioned already HMOs are these non-nutritive sugars that support the growth of bifidobacteria, but they also help to reduce pathogenic, nasty, non-healthy bacteria such as staph. And they also are antimicrobial to organisms like group B strep, which is, again, a rare but important infection in newborn babies. So these HMOs are more than just food for bacteria. They actually have immune functions as well. And the diversity in the gut, that range of different bacteria, is very important in developing a well-functioning immune system. And we see there is lots of evidence that in newborn babies, as they developed, the maturity of the immune system is important in not just fighting off infections, but also educating the immune system as to what's normal and what's not normal, and therefore reducing the risk of autoimmune conditions, but allergies as well. So the immune system is not just about fighting off infections. It is responsible for some important diseases in childhood and later on into teenage and adult years as well. The diversity is also important for metabolism. So metabolism in bacteria is the fermentation of nutrients, and this is where they produce these short chain fatty acids, SCFAs that I mentioned, butyrate being arguably the most important of them, and they produce lactic acid and other metabolites, which are, again, really important for developing a mature gut. The intestinal barrier is something that stops bacteria leaking out into the bloodstream. And so a leaky gut is associated with the development of bacterial infections. And again, these healthy metabolic functions of probiotic bacteria reduce the likelihood of that. And they help to resist colonization by pathogens. So pathogens are bacteria that are not good for us. And they actually actively produce compounds that can kill gram-negative bacteria. So you may have heard of things like E. coli or Klebsiella, which are nasty gram-negative classed bacteria that, if they get into the bloodstream, can make people very sick at any age. And again, the diversity of a healthy gut microbiome helps to reduce the prevalence of those gram-negative and other bacteria. And essentially, they seem to have direct effects on them by producing these things called bacteriocins that kill them, but also just in numbers, they compete and can outcompete them for nutrients as well. So again, diversity is hugely important to reduce the prevalence of those harmful bacteria. And then I think something that is very relevant, particularly for this talk, is the promotion of neurodevelopment. And this is something that, again, over the last few years, there's been a lot of research into. And this is the so-called gut-brain axis, where the diversity of the gut seems to have a direct influence on neurodevelopment, and the metabolites produced by bacteria have a direct influence on brain development and brain function. And of course, particularly in the developing baby and child, then we're talking about neurodevelopment. We're talking about having the right compounds in the right amount, helping the brain to develop in a normal way. And we also know that having the wrong bacteria so things like Klebsiella that I've mentioned, but Streptococcus is another one. There is evidence that where they're present in abundance, they're negatively associated with gross and fine motor scores. In other words, they're having a negative effect on normal neurodevelopment, whereas Lactobacillus in abundance has a positive association with normal neurodevelopment. Now, these at the moment are observations from human studies and from research studies, but these are incredibly important findings, which really go a long way to explain some of the things that we've seen over the last few years, and imply that there are windows of opportunity to actually modify some of the problems that we see at the moment. So moving on now to some of the immediate and longer term effects of a gut dysbiosis. A stable microbiome appears around the age of 3 to 4 years old. So in those first 3 years after birth, this appears to be a critical window while babies are developing their metabolic, their endocrine, their nervous, and their immune functions. And programming that microbiome and the microbiota, which is the genetic part of it, may well reduce the risk of developing diseases later on in life. And these are influenced by what the unborn baby is exposed to during pregnancy, and also the health of the mother. But what we also know is that the gut microbiome seems to reach a stable adult-like state by about the age of 3 to 4 years old. And after that, it doesn't really change very much. So the argument for normal healthy adults to take regular probiotics doesn't really exist, because you can't really do much about it. If you don't have a healthy gut, then there is an argument for having probiotics. And if you have a disrupted gut, reflux or antibiotics or gastroenteritis, then there may be an argument. But the really important bit is stabilizing the baby's gut and making it healthy as the baby moves through that transition from being milk fed to food fed, and getting that big diversity of food into babies. And the first 3 years appear to be critical in achieving a stable microbiome. And if you don't have a healthy microbiome by that age, you may not be able to get one very easily at all. It's a really interesting hypothesis, because this then may define what happens to you in later life. And this is where lots of interesting work saying, well, if you don't have a healthy microbiome by the age of 3, you're at risk of developing, for example, obesity and type 2 diabetes, and it's slightly out of your control, which is A, a little bit terrifying, but B, again, gives us the opportunity to think that some of these things we may be able to modify and influence. So what happens straight after birth? Well, as I've said, the neonatal gut is rapidly colonized by bacteria. The bacteria consume oxygen. Babies who are a bit more premature have less diversity, and the mode of delivery results in a different bacterial balance in the gut. So we've covered those things. And we know that breast milk favors some of those healthier bacteria compared to babies that are formula fed. And we know that babies who are formula fed have a slightly higher abundance of bacteria that we would consider to be harmful, like E. coli and Clostridium and Bacteroides. The names don't really matter. But again, it's trying to get that healthy microbiome established. And we know that some of the things that we do in the neonatal period disrupt the development of a healthy microbiome. Over half of babies now are born by caesarean section, and there are good reasons for that, because one continues to drive down risks to mothers and improve the safety of delivery. But at caesarean section, unborn babies are exposed to the antibiotics that are given to their mothers. And those antibiotics hang around for a couple of days in breast milk. So babies start life with a fairly significant exposure to antibiotics. And we know that that exposure can persist in the gut for the next 2 years. Some babies are given prophylaxis, so antibiotics because of risks of group B strep. Again, very sensible, because group B strep can be devastating in babies. But again, the effects of even a single dose of antibiotics can be seen over a year later in babies, looking at their stool and the microbiome. And if mothers who are breastfeeding are given antibiotics, again, that exposure to the baby through breast milk can have an effect. And again, there is evidence of a long-lasting effect on the microbiome. So these well-intentioned interventions, which are done for perfectly good reasons, and we shouldn't necessarily not do them, but we have to think of the consequences for the baby and perhaps what we might be able to do to mitigate and modify the impact of those effects. So just to give you an example of a study, this was a study of early antibiotic exposure in 212 babies overall, and 45 of them were exposed to antibiotics. And those who didn't get antibiotics were less likely to develop allergies, less likely to develop fungal infections, and had better growth as well. So that's just one of, dozens and dozens of studies which show these clear effects. There is evidence that antibiotics given to babies can cause poor growth, and the more antibiotics you give, the worse it is. And that seems to be particularly around bifidobacteria. Bifidobacteria is very sensitive to antibiotics. But we also have observations that if you don't grow well in childhood, that is associated with neurodevelopmental problems, but also cardiometabolic problems like obesity and type 2 diabetes in later life. So there are strong things that are linking events that happen very, very early on in our lives to things that come to affect us later on. This was a big study done in the UK of over 3,300 babies, where they looked at serial stool samples. And they essentially were able to categorize the bugs that they found into 3 clusters. And babies born by vaginal births had 3 clusters mainly, and babies born by caesarean section had 2 clusters. And they found that cluster 1 was a sort of mixture of species, cluster 2 was mainly bifidobacterium, and it didn't matter the mode of delivery. But cluster 3 was mainly Bifidobacteria longum, specifically that bug, which was only found in vaginally delivered infants. And they followed these babies up for a couple of years. And what they found was that the babies who had the cluster 3 were 3 times less likely to get viral pneumonias than babies with the cluster 1 pattern. And viral pneumonia admits, children get admitted to hospital with bronchiolitis and other nasty lung infections. So again, this is very much related to the diversity in the gut and the protection that gives to the immune system. And as I've mentioned, the persistence of changes that we see in babies, this idea that they can be exposed to antibiotics and a year or even 2 years later, the gut hasn't returned to normal, that isn't seen in adults. The adults bounce back to that pattern of the microbiome when they were 3 or 4 years old. So this suggests that there is a unique and a critical window that exists in those first few years that might enable us to stabilize or correct and achieve a healthy microbiome in young babies, which is a window that may not be available to us later on. Okay, so what about specifically in neurodevelopmental disorders? So there are some important questions to which I mainly don't have the answers. Does a dysbiosis affect or contribute to the neurological development or to symptoms in children with Down syndrome? That's a really important question. Are there any particular dysbioses, types of dysbioses that are more common in Down syndrome? Is there any evidence for the benefit of probiotics in people with Down syndrome? And are there any additional safety considerations that we should be thinking about if we're thinking about giving probiotics in Down syndrome and other neurodevelopmental disorders? Okay, so what do we know? Well, what we know is that first 1,000 days, first 3 years, that's a nice convenient number, but disruption of the microbiome in the first 1,000 days coincides with key stages of neurodevelopment. And there are animal models that are bred to be completely free of microbes. And in those models, what people have shown is that where there is no microbiome in early life because they're sterile, those animals develop neurodevelopmental abnormalities. They become stressed, they become anxious, they develop abnormal social and emotional behaviors, and their brain function is affected as well. And if they are then given specific bacterial strains, if they are given probiotics, they then develop positive behavioral and neurochemical changes in the brain. So these are things that you can't do to humans, and much as we'd probably rather not do animal experiments if we didn't have to, we can certainly learn a lot from these very tightly controlled environments. But after 1,000 days, restoring those functions becomes nearly impossible. So you can restore the functions in the first 1,000 days, but after that, it seems to be much more difficult. There is also evidence that an altered microbiome in the newborn is associated with an increased risk of some common neurodevelopmental disorders such as ADHD or ASD. And that those are associated with lower levels of myelination of the brain. So normally the nerves of the brain, the neurons, are covered with a sheath of myelin that massively improve the speed of transmission of nervous symptoms. So myelination is a really important thing. And therefore, if you have less myelination, your brain is not going to function in the way that it should be able to function. But again, in some animal models that have been created with ASD, when they're given lactobacillus, you see a reduction in those symptoms as well, implying a direct effect of some of the metabolites that are released by the presence of lactobacillus. And of course, there are lots and lots of causes of dysbiosis that aren't just present in children or adults with Down syndrome. These are risks for all of us. So maternal influences, newborn influence, mode of delivery, exposure to antibiotics, feeding practices, and I've covered those. But what we do know is that longer term dietary habits help to create a stable microbiome, and some dietary so-called treatments, and I put it in inverted commas deliberately because one has to be very careful, things like the ketogenic diet that people have heard of, can significantly change the microbiome, but not always in a good way and unpredictably. So one has to be very, very, very careful indeed. Medications can cause dysbiosis, and not just antibiotics. Things like proton pump inhibitors like omeprazole changes the pH of the gut. Even a bit of Gaviscon can change the pH of the gut, and psychotropics can as well. Of course, your genetics, which you can't do much about, can influence dysbiosis, and your immune system. It can also cause dysbiosis as well and be influenced by dysbiosis. So some of these things, again, seem to play on each other. And again, this is a diagram which really shows a number of effects. So you've got the gut wall here, and you've got some of the bacteria, and then you can see some of the effects on the brain, on the immune system, on the endocrine system, on the nervous system. So this is really just summarizing some of the things that I've mentioned. So just into the last bit, what do we specifically know about the microbiome and Down syndrome? And again, the answer is not enough. But we do know that in people with Down syndrome, there is often significant dysbiosis with reduced diversity and shifts of microbial colonies away from healthy guts. There is a greater tendency to gut inflammation and a vulnerability to gastrointestinal symptoms like constipation. There are neurodevelopmental complications, including ASD, and studies that have looked at profiles of those short-chain fatty acids like butyrate have shown that the profiles are not normal in people with Down syndrome, and that there are disruptions of neurotransmitter signaling. Down syndrome seems to be specifically associated with a dysbiosis where lactobacilli and bifidobacteria are often reduced. And as I've said, those are critical in maintaining a healthy gut and an immune aware gut. And therefore that leads to an abundance of pro-inflammatory pathogenic bacteria. And there is a really close association with gastrointestinal issues in about 60 to 75% of people with Down syndrome. So the gut and the condition are closely linked. And again, in animal models, there is evidence that dietary changes can influence the dysbiosis, again, either positively or negatively as well. And that gut dysbiosis is linked to processes such as inflammation and oxidative stress, which are things that the human immune system uses to try and regulate itself against infection or against attack. But those also have negative effects on brain development, and those have negative effects on IQ and on behavior. studies have measured the production of SCFAs in Down syndrome, showing that there is a significant reduction where the underlying condition is present, especially butyrate. And a lot of this is supported by good models of animal evidence, but the human studies are still lacking. There have been so-called interventional studies where if you change the microbiome in animal models of Down syndrome, you can reduce ASD-like symptoms. Bifidobacterium infantis in particular has been shown to restore the microbiome and improve social behaviors in animal models of Down syndrome. And a lot of the current trials are really focused on the first 1,000 days as key windows. But what we don't know is whether other windows or opportunities exist. They just haven't been studied. So we just don't know. And that's a real shame. There is limited evidence of what we should be doing and thinking about in terms of probiotics and dysbiosis in people with Down syndrome. We don't know what the optimal timing or duration of giving probiotics or other interventions should be. We don't know whether some probiotic species might be more effective or not. We've got things like Bifidobacterium infantis and Lactobacillus showing promise, but nothing beyond that. And again, what doses might be effective and whether individuals might respond better to certain interventions, others, again, an awful lot that just simply isn't known. So, to summarize, and as I said at the beginning, whilst many of you will have family who have Down syndrome, you will also have family who don't. So what I hope I've shown you is things that is relevant for everybody, because I think it is relevant for everybody, and it's poorly understood by doctors and nurses, never mind poorly understood by people who aren't medical. But gut dysbiosis is really important, and it's really common, and it's poorly recognized. It starts at or before birth. It has long-lasting effects. It seems to impact on human health at all ages, and it is a global health challenge. And this is because the gut microbiome is so important in the development and function of the brain and nervous system, the immune system, including the development of allergies, metabolism, and other problems in adult life, including colorectal cancers and others. And we know that Down syndrome is associated with disruption of that gut-brain axis, and it is quite possible that a healthier microbiome could at least reduce some of the neurodevelopmental issues that are seen in children with Down syndrome. And an early focus on the gut microbiome, particularly under the age of 3, is currently thought to be the optimal area for further research. But as I say, there may be other windows that we just don't know about where we could manipulate the gut in a positive and long-lasting way and see some benefit. But the good news is that probiotics are safe. Millions and billions of doses of probiotics have been given, and they appear to be a remarkably safe intervention and seem to have a useful role in altering the gut microbiome in a positive way. But I'm afraid the current state of play in 2026 is that significant knowledge gaps still exist.